So today we're going to talk about the structure of DNA and RNA as genetic material. DNA and RNA were first discovered back in the 1800s, and they knew that they were had different components, and they knew that they were found in the nucleus, and RNA was sometimes found in other parts of the cell, but they didn't really know what it was and what it did. And they didn't even know for a long time, even after they started working out the science of genetics, what it was exactly that carried the genetic message from generation to generation. So all of these things were things that had to be worked out. The genetic material for a long time was considered to be protein, probably, rather than something like DNA, because proteins could be so large, and there were 20 different amino acids, and they could be put together in lots of different ways. And so for a long time, people were trying to figure out what it was actually that caused or played a role in heredity. One of the things that led to the discovery of DNA as the material that makes up our heredity was begun in the field of molecular biology, where people were studying bacteria and viruses. And eventually that led to the discovery that DNA was the material that was involved there. In 1928, a scientist named Frederick Griffith first discovered that there was some kind of transforming factor that could be transferred from one bacterium to another and then inherited by future generations of bacteria. He was working on two different strains of the bacterium that is most commonly found in pneumonia. One strain causes pneumonia, and the other strain, which is the same species, doesn't cause disease. And so he was doing various experiments with those, and one of the things he did was he took some of the known disease-causing bacteria and killed them with heat, and then put them in the same growth medium that he had some non-pathogenic or non-harmful bacteria of the same species. And lo and behold, the non-disease-causing bacteria were transformed or changed into disease-causing ones. And so there was something there in those dead bacteria that could change the genetics, change the inheritance of future generations of bacteria. He called this transformation because they were transformed from harmless to harmful in a very simple way. So scientists worked on this for quite some time to figure out what it was that caused that. There were various experiments that were done, and over the next 15 years or so, a lot more evidence was found to point toward the fact that it could have been DNA. The ultimate experiment that was done to prove once and for all that DNA was the transforming material was done in 1952 by Alfred Hershey and Martha Chase. They used a type of virus called a bacteriophage to show that DNA was the genetic material of the virus. Viruses are made of a nucleic acid like DNA or RNA and a protein outer coat. Proteins have sulfur commonly, but they don't have phosphorus, and DNA, of course, has phosphorus. And so they made viruses that had radioactive sulfur in their protein, and other viruses that had radioactive phosphorus in their DNA. And they tested both of them with bacteria and found that bacteria that were affected had the radioactive phosphorus inside them, and not the radioactive sulfur. And so that was kind of the definitive experiment to show that DNA was the material that did this. This is a picture of a T2 virus, similar to what Hershey and Chase were working with. It basically looks like a lunar lander. These little tail fibers here are not really legs. They kind of look like legs, but they do not walk, they do not move. They're just used for attachment on the surface. So DNA and RNA are the two nucleic acids, and so let's talk a little bit about what they're made of. One of the two strands of DNA is called a DNA polynucleotide, meaning that it's a polymer of nucleotides. Nucleotides, as you recall, are made of a nitrogen base, a 5-carbon sugar, and a phosphate group. And then the nucleotides are joined together by a sugar-phosphate backbone. The sugars and phosphates are bonded together, and the nitrogen bases are attached to the sugars. Each type of DNA nucleotide has a different nitrogen-containing base. Those are adenine, cytosine, thymine, and guanine, as we've talked about before. and they're put together like this in a chain. So you have here a chain of sugars and phosphates, sugars joined by phosphates, and then the bases are attached to the sugars there. These are a couple of different representations. This one is just showing a basic ribbon diagram and this one is showing a modified structural formula there. And then here we see the structural formula of a nucleotide. This one happens to be a thymine nucleotide. That shows you how they're connected, and then the phosphates are going to connect one sugar to the next in the chain as it shows here in the middle diagram. These are the 4 nitrogen bases: thymine, cytosine, adenine, and guanine. Thymine and cytosine belong to a class of chemicals called pyrimidines. They have a single ring structure, 6-carbon ring structure there, with a couple of amino groups attached or part of the structure. And then adenine and guanine belong to a group called the purines, and they have 2 ring structures, a hexagon and a pentagon, that are attached together. And these are called the purines. These are called pyrimidines. RNA is different than DNA because it uses ribose instead of deoxyribose, and it has uracil instead of thymine. It's also usually only a single strand rather than a double one. Here's a uracil nucleotide similar to the thymine one we saw earlier, except it does have ribose sugar rather than deoxyribose and uracil rather than thymine. This is a diagram of an RNA molecule showing you that it's a single chain there of the different bases that are linked together on the sugar-phosphate backbone. So in 1953, Watson and Crick were the ones that finally figured out the secondary structure of DNA. There were a number of scientists around the world that were working on this, trying different kinds of methods to figure it out, biochemical methods as well as other kinds of physical methods. Two other scientists in England, Rosalind Franklin and Maurice Wilkins, were working on a technique called X-ray crystallography, because DNA can be crystallized, kind of like sugar when you remove the water from it. And what they found, the picture that they took of the crystallography, the X-ray of the DNA crystal, led Watson and Crick to make their final deduction about the structure of DNA. They also used another scientist's information. Chargaff had observed a number of years earlier that in DNA, the amount of thymine always equaled the amount of adenine, and the amount of guanine always equaled that of cytosine, and so that led them to understand something about how the bases were paired up. This is a picture of Rosalind Franklin, and this is the famous X-ray photograph that she took of the DNA double helix crystallized, and this shows that that double helix, if it's twisted around like that twisted ladder structure that we're so familiar with, and you look at it from the end, from one end or the other, it makes this X structure in the middle. And once Watson and Crick saw this diagram, they knew that there had to be a double helix involved in this structure. They reported that DNA was made of 2 polynucleotide strands that were wrapped up into a double helix with the sugar-phosphate backbone on the outside and the nitrogen bases perpendicular to the backbone on the inside. The adenine would always pair with thymine, and cytosine would always pair with guanine, and that would give it a uniform diameter all the way down the length of the molecule. It's not necessary for you to know this, but adenine does form 2 hydrogen bonds with thymine, and guanine does form 3 hydrogen bonds with cytosine, which lets you identify which one is which when you see them in a structural formula. Here's a picture of Watson and Crick with their model that they built using various structures, clamps and ring stands, and wire and cardboard pieces. You can see in the picture that Watson is very young. He was about 23 or 24 at the time, and Crick was in his early 30s when they made this discovery. So what they basically said was that it was like a ladder that is twisted. The side pieces of the ladder were made of alternating phosphates and sugars, and the nitrogen bases formed a pair that made the rungs of the ladder, and then if you twist the ladder, that gives you the double helix structure. And here's the basic diagram that shows you all the different ways you can look at DNA. This one is called a ribbon model, and you just see it shows the the backbone is as a ribbon of alternating colors with sugars and phosphates, with the base pairs forming the rungs of the ladder. This one is a partial chemical structure showing you the arrangement of the atoms and the molecules together. It shows the hydrogen bonds between them. And then finally a computer model that shows the space-filling structure. The Nobel Prize was awarded finally in 1962 to Watson Crick and Maurice Wilkins, one of the X-ray people. Rosalind Franklin probably would have been recognized as well, but she died from cancer in 1958 due to having been exposed to X-rays during her years working with X-rays. But their model allowed us to learn more about how genes and chromosomes work, because the genetic information on a chromosome is encoded in the nucleotide sequence of DNA.